I will talk about something that some of you are familiar with. That is what we called yesterday and what we call now a microbiome, a new word in the microbiology. So it's all about microbial ecology, that means different groups of organisms working together. And then, since we are applied, to bring some new technological perspectives. Now my main story, I will deal with some basic principles which date from very recently in microbial ecology. Things that we observed, that we got underpinned by recent publications, which now stand, and which we can build on to deal with microbiomes, with these organized groups of organisms, to do something. But as my talk goes on, you will see that many of these things which we find now back in microbiology are actually applicable to our society, to our market economy. And inversely, some of the things that we observe in our market economy, you can apply to our microbial ecology. So this will come through my lecture back and forth, and it helps us to understand microbial ecology and technology, and it might also help us to deal with some of our issues in our societies. I will talk about, first of all, what is a microbiome, as we understand it now. What is it, and what does it mean in terms of operational concept, of engineering concept? What is the methodology? I will speak a little bit about the interfaces where we find these microbiomes and where they do add information and communication technology. I will then talk a little bit which of those manufacturing microbiomes we are particularly looking forward to use in our society. And I dwell a little bit on that, so I become a philosopher here in number 5. And then I look a little bit ahead in the future in number 6. And I have some conclusions for you to take home. A microbiome is a real team and not a haphazard mix. Lots of people thought, oh, some organisms fall together, they do it. It's heuristic, it's by probability. It has been shown it's deterministic. It happens and it's constructed, it's a team. Now, how do you know you have a real team? The story is that you do a Pareto analysis. That means you look for the 20 most important organisms in your microbiome, and if these 20% do 80% of the job, you have a microbiome. That's the real world. Any company, everywhere in society, 20% of the people have 80% of the money. Right? That's Pareto. That means you have a microbiome. This means also that 80% of the people or 80% of the species which you have in your group there, in your microbiome, are just watching for a moment to jump in and to take over. They keep those 20% very active. That's the meaning of the 80%. Now, you will also observe that in a microbiome there is a lot of cross-feeding. An organism does task A, could do B, but gives it to another organism, because that other organism can do it better. It gets more exergy, useful energy per delta G, per unit of free energy change. And you get what we call slow metabolism. A microbiome, everything goes slower because they want to make the most out of it. And you will find that diversity increases, and as diversity increases, stability increases. A microbiome is constantly changing to new traits. Now we know that there are heritable phenotypes resulting from distinct genotypes in plant communities. Well, this has been established. For microbiology, it has not been established so clearly, but it is happening. That means that the microbiome constantly develops new novel genes and, which is very important for us, new products. Dealing with microbiomes is dealing with something which constantly explores new things. Never be surprised. It is a fact. It happens. This is very bugging to those who make patents. They want to describe this and they can never put this, a microbiome, in a patent system. Now, these generators of new life, of new traits, are not the generalists. We now know this. It are the specialists. The specialists have a smaller genome, and they constantly focus for fine-scale niche participation. So microbiomes are generators of new value, and the specialists are the drivers for those new niches. We very much talked yesterday, a number of you, about protection, health. Well, it has been established now for the first time that the best protector of our health in society and our environmental system is biodiversity. The more biodiversity, the less disease. The established microbiome has a very strong barrier function. Microbiomes protect against invasion. And instead of fully focusing, I'm not against antibiotics, but instead of always focusing on antibiotics and disinfection, we should learn to much better explore the barrier function of microbiomes. They are all over us, on our skin, in our gut, in the rumen. They protect us much more than antibiotics. Antibiotics have to be the exception of the exception. The microbiome is our protector. This brings me to the concept of methods. Of course, we deal with genomics, but genomics in microbiomes doesn't help us a lot. They give us lists and ratios. This is more important than that. We can see if we have Pareto. That's okay. But basically, when we deal with microbiomes, we need to think about system genomics. And system genomics are complicated. We need bioinformatics for that. Now, if you deal with microbiomes, looking at a gene is like looking at a machine in a workshop. It doesn't tell you anything of the output of that workshop. It's like looking, if you look at a genome, it's like looking at a factory. But a factory is part of a whole society and can be part, and that is the level of the microbiome of a multinational. Looking at the microbiome is seeing what is the multinational doing. A microbiome is the representative, the corollary of a multinational. So that's what we like to know if we deal with microbiomes. What multinational is driving the business in that system? Of course, the nagging question is, if I have a microbiome, my gut, the rumen, my skin, an activated sludge, a fermenter with mixed cultures, where is it going? And that's a headache. That's why people like pure cultures. Because that headache is there, what can happen? And that is where we still have problems, and what we are now doing is not looking at the catabolic genes, but looking more at the anabolic genes of this microbiome, because they can signal where the microbiome is developing new intelligence. For instance, how does it, when are certain spores desporulating and things like that. We have to be able to predict where it is going, otherwise it's difficult, but that is the level. You will say, how do I get a microbiome? How do I get such a team? How do I get such a group of organisms performing well? Of course, you can start with scratch, and this is now where we are. A number of people are trying to make new microbiomes. For instance, you all know these cultures, these pure cultures, to drink, Actimel, to name one. That's one or two organisms, but they are defined. Now people are defining or trying to develop microbiomes to take in. Now, they start with some generalists, and then they will try, by genome editing tools, to add to those generalists certain particular traits with a priori in silico validation of what has changed. So this is the future of the microbiome for very medical configurations. But at this moment, we are not, I know of the first attempts in that line, but now at this moment, engineering a team starts with what we call, you take an existing microbiome. You take some part of the rumen, you take some part of the soil, you take some part of our gut, and then what you do is you take that existing team and you start to massage it, you drift it, you bring an accelerated evolution. As it has been millions and millions of years evolving, you push it in a particular direction. For instance, in the environment, this is one example. These people could make a microbial team which was actually a group of organisms, and they can stand very fluctuating levels of ammonia in biofilters. Or some people are now working on vaccinating microbiomes so that they can resist invaders like viruses. So the rule of thumb is, first, use some knowledge. Do I have a microbiome? Do I have an organized team? Is there Pareto? Is there cross-feeding? If you have it, implement microbial management to create new traits, new knowledge in that team. And when I say management, I say information and communication technology. Indeed, a microbiome, a multinational, is all depending on constantly sending messages about their three-dimensional structure. They are in 3 dimensions organized. You sit there, I stand here, and you do that. And I have to be in that order. And they organize in 3-dimensional way. We know this now for certain examples. For instance, we know if you take a methanogenic consortium that grows on propionate, well, it is very well organized. It's very nicely situated. So how do they do that? They use things like pheromones, AHLs, nanowires. I showed yesterday a picture of it, electronic shuttles, gradients, and even patterns of advective flow when they are in flocks. They grow in flocks, in biofilms. And the way that the water flows through a flock is like a river going through or a street going through our suburbs. That's the way they organize. It's all very human. You recognize it. You look around you and you transplant it to the concept of how microorganisms organize. What are the sides of interactive microbial behavior? Well, they are the interfaces with other things. For instance, you have external cohabitation, which we know, the rhizosphere of plants. Plants grow by the blessing of bacteria. They have rhizosphere. The philosphere on their skin. We grow, we have more microbes, microbial cells than we have human cells. Our skin, on abiotic materials now, there is a lot of development of growing organisms, groups of organisms on electrodes. There, these microbiomes feel very much at home, at these interfaces, not suspended in the water, not planktonic, but on interfaces, microorganisms grow to densities of 40 grams per liter, like we do in our mega cities. That's where they grow. That's where they are. And you see that in the case of a higher host, for instance, at the mucosa, you see that the higher hosts then help these microbiomes. For instance, they change, they have immunoresponses towards those organisms which are very close to the host. They have actually particular privileges to those which they trust, to those which are subdue. So the same happens in roots. Roots have organisms which come very close to the organism, to the real nature of the plant, and then the root even supports this. So it has been shown even that for roots, that the interface, the root microbiome, actually can be transplanted. So we can take a root and transplant it to another place, and that microbiome is taken along with the root. And this is a tool which we can use, and I'll give an example further on. A second type of interface is where we get even closer, where we have mutualism and symbiosis. For instance, the root nodule. A nodule is an internal structure. The amoeba legionella vacuole, the Wolbachia insect system, the mitochondria, which have some organism particularly in them. Here we are working. The microbiome concept in such interfaces is still developing, but I predict that we will have root nodules in the future which make plant defense chemicals or vitamins or special bioactive proteins. And that we might, and I dream for those of you who know that rumen exudes so much methane and is part of our climate change, we probably will have pretty soon mitochondria developed which contain in the rumen mitochondria cells, which actually contain methanotrophs, which consume then the methane, or which contain butyrate-producing bacteria, which then make something useful instead of that methane. This brings me to the point of manufacturing microbiomes. Manufacturing is extremely important in our society. Where now do we need microbiomes, and where can we continue to have pure cultures? And where do we use just plain chemistry? That's the question. We don't have to use microbiomes where there are fierce competitors. So I see 3 areas where we will further need, or where we really will need microbiomes. The first is the total dissipation of substances at minimal cost. We call this cleaning up environment. We want molecules, pollutants to be degraded, right to the right little atom. This, of course, has been developing over the last 20 years, but now we see that there is a focus not so much on total dissipation, but also on recuperation. But that is a domain, the environmental microbiome, to clean up the environment of pollutants, of everything. The second domain is make molecules of a higher order that not the chemistry can make or that even pure cultures cannot make. And there are good examples now coming, and I'll give an example. Single-cell proteins, polyhydroxyalkanoates, vitamins, exopolysaccharides, which are only formed under combined conditions where A, B, and C work together. And the third area is making a healthy entourage. For those of you who don't understand French, it is making, surrounding us by a healthy group of organisms which are our partners and which keep us alive, which keep us counteracting those organisms which are loaded with all kinds of factors which can make us make us sick. So we tend to always, when we talk about homeostasis in dealing with things like pandemics and antibiotic-resistant genes, we never talk about the potential that is in the microbiomes that are around us, on our skin, in our gut. We should much more look at that in the future. Oops. This brings me to the microbiome and some philosophical considerations of the society at large. And I'll have 3 points, some lessons in general, some planetary challenges, and the market economy. If you look into the literature these days, you see people talking about a minimal life. That means how can we survive? What are the minimal conditions? In deep soils and sediments, in deep oceans, we find communities which thrive with minimal entropy and maximum homeostasis and slow metabolism. This is something that we should explore. We can learn a lot about it. How can we do it here on Earth? We, human beings, we can take an example here. In oceans, I was always puzzled. There are 10 times more viruses than there are bacteria. And I was always puzzled how that come. The answer is the viruses drive the recycling. So we always talk, we have to have a more recycling in this world. Well, here is a role for viruses, I think, and we have to integrate them in some of our biotreatment processes where we deal with recycling. And this is for people like me, old age people. When we look at older cells in microbiomes, when they get damaged, we then see that they do something very strange. Well, very strange, very logical. They transfer all their good traits in a daughter line. So they get eternal life. Now, isn't that a morale for all of us? And we can learn from how microbiomes take their own resources and instead of letting them go lost, they transfer them in the microbiome to the next generation. So you see that our society, there are boundary conditions here from which we can learn in terms of basic needs, optimal cycling, and methods to activate the next generation. Beautiful things to learn. I give you an example in terms of planetary challenges. You all know that the CO2 business is in. And everybody talks about these Ferraris who make so much CO2, our cars. Actually, the amount of CO2 made by the soil microbiomes is 10 times that of made by the transport sector. And we don't do anything about this. We don't even know how to deal with it. So some of you know that in the last 10 years, 10 years ago, nobody wanted to work about the gut microbiome. This was shit, right? Nobody wanted to work about this when I started about that. It was the dirtiest thing. In the lab, I had to literally take somebody and force him, you know? In those days, it was still possible. Now you couldn't do this anymore. But 10 years ago, and now, at this moment, nobody else works on this, and it will come. It's the way to deal with climate change, because this microbiome, we must learn to control it and to deal with it. And I'm quite convinced if we change it a little bit and learn for certain agricultural systems to deal with it, we resolve a lot of the problems of CO2. That brings me to the market economy. Here is an economist who says, we may have future financial crashes if we view the economy like an ecosystem. Indeed, The economy is like an ecosystem, and the ecosystem has its economy. For instance, we talk a lot in the economy about the concept of minimal guaranteed income for each. Or in Europe, we talk about what is called the Morris theorem, that the world dominance of the Western culture, don't be offended, it's a theorem, is due to exposure, challenge, and change. We can test such concepts at a million individual scale in a microbial system, in a microbiome. So these are things we can learn where economy and ecology can be dealt with at the level of the microbiome. And I gave this slide yesterday for those who are here. It has now been established. You learned a lot, you all heard about unculturable bacteria. What were unculturable bacteria? Bacteria which sit there alone and say, this is no life, this is no fun. I can make this, but I give up. Unculturable bacteria don't like to do, and those bacteria who grow alone are really, they are unusual. Most of the bacteria immediately start by dividing the tasks and by being better off, by having a much easier life. And it are economists, actually, who now developed these models and clearly proved it. Exchanging, trading metabolites between species, multi-species, as it happens in a microbiome, gives a much easier life to microbes. So lots of those unculturable bacteria are bacteria who just get scared when they are alone. They are not unculturable. You give them a partner and they're happy. And they do it. Some key microbial ecology elements that we still don't know much about. In microbial ecology, we have organisms which grow very quick and those which grow very slow. They are progressive and conservative investors. That's the corollary. We don't know how to deal with them. We have team players, but also in microbial ecology, you have profiteers. Do you understand the word? You have organisms who never deal with it, just sit back. The same genotypes, there are phenotypes under the genotypes, we just sit back. And we have now learned, we've got pretty good evidence now how to avoid that. By changing environmental conditions and having good gradients, you take out the cheaters. You have sexual promiscuous versus Puritan bacteria. Sexual promiscuous, these are the horizontal gene transfers, and they are in hot spots. I don't have to define hot spots in societal terms, I think. And there is the Hubble niche theory, a theorem who says everywhere where you have an organism, it makes itself finally a kind of niche, a place, and a business, so that it's happy, thereby improving its own fate. This means that when an organism arrives somewhere, pretty soon later, it makes its own environment, its own niche, and it becomes pretty happy. All of that is something that we are learning, but have not been able to implement so much when we deal with microbiomes. Some platforms of progress to finish. Well, very soon we will have the so-called gene drive systems, and that will help us a lot to actually manipulate microbiomes. But we will be able to have improved guidance of DNA functioning of some of those species, and we will be mapping in advance the outcome of the functionality in this open system. So I predict that microbiomes in the future will give us new unexpected microbial outcomes and opportunities. It's wisdom of millions of bacteria together, and we will add our wisdom to it through these gene editing systems. We particularly will be dealing, I think, in an area for the future at these interfaces of higher and lower organisms, bacteria and plants and algae and insects and animals. For instance, we will be able to look at having lower levels of antibiotic resistant genes. This is something that we have and can deal with when we look at the microbiome. Have a less immunodeficiency, have a better immunoresponse to organisms, better microbial entourage, protection against invaders, using competitive exclusion instead of disinfectants. It exists and it is already done for biocontrol. And having microbiomes, it's all in the same line, vaccinated against X, Y, and Z, so that they help us to outlock some of those organisms that we don't want. An example that I kind of like, I hope it's underpinned in the meantime, This is an experiment. This was a new scientist, and the story said superplants may beat the heat. And the idea is simply that one took an endophytic microbiome, which is in the plant, of a very heat-tolerant plant, and they put it in wheat. And the wheat was capable to grow with 50% less water and could endure 70 degrees. And there was not a genetic manipulation, it was just a transfer of a microbiome from one plant to another. This opens a lot of perspective, I think. Transferring microbiomes with all their wisdom from one situation to another situation, and then have that capacity transferred into for better use of us. And that brings me to my last point. We are in our society dealing with a cyclic economy. We have to change our way of living. We now bring through massive transport from the agro environment things to our mega cities. The mega cities are constantly getting bigger. And we have this massive transport from all over the world to those mega cities. And when it's there, we use and consume it, and then we have to destroy it. And to destroy it, we should now start thinking to recycle it. And to recycle it, we should have these empowered microbiomes to upgrade our race. Not destroy them, but upgrade them. Yeah, how do we do that? Well, I think we must think that when these things arrive in our cities, we consume them, that we should also make, again, feed and food from it. And I give one example for that on which we now work, and that's the nitrogen cycle. And the nitrogen cycle, the last slide, is then, you may know that we now all use Haber-Bosch industrial nitrogen to make our feeds and foods. Haber-Bosch consumes 2 to 3% of our total energy. All fossil fuel in the world, 2 to 3% just goes for one thing, making industrial fertilizer. One thing. When it comes to the land, this massive amount of nitrogen, the efficiency of our crops all over the world, worldwide, is not more than 50%. So 50% of this nitrogen goes in the air or comes in the groundwater. And some of you may know that number 3 of the challenges for our planet is the nitrogen pollution, the nitrogen polluting species. This is the challenge number 3 of our world. It's part of climate change. So we are here in a very wrongdoing. When this food then comes to livestock production, you have an efficiency of 10%. And when it comes here, you have another efficiency of 10%. So you are at 1% of, here is 50% of the nitrogen lost, and here 1/10 comes here. And when it comes to our sewage treatment plant, we destroy the rest. So this is a totally ridiculous story about nitrogen. And we have to do something else. And this is a paper that you can find as a cover issue of Environmental Science and Technology recently. And the story is that we say, look, we must take this nitrogen which is lost here, we recover it, and we use now we now produce, as it was in the 70s, co-chair, single-cell protein. We use microorganisms growing on organic fiber, microorganisms growing on hydrogen, microorganisms growing on even green energy, electricity that we generate from photovoltaics, and we grow and we make single-cell protein again. And similar here, instead of destroying all this nitrogen, we use microbiomes who use the nitrogen and then together, because single-cell cultures are not so productive, but microbiomes, teams of organisms can directly make use of this ammonia and reintegrate it into single-cell protein that we can consume, us as human beings, and we no longer need this massive amount of nitrogen going into our environment. So much about microbiomes, some conclusions in 5 sets. A microbiome is a team, it's a multinational. It uses maximum exergy, it tries to get the maximum useful energy and needs maximum complexity, needs a lot of knowledge. Its operational hallmark is Pareto. Strangely enough, 20% are active, 80% are waiting to get active, but those 80% are important because they keep the 20% really eager to continue. It's like in football. You don't perform on the back. The trainer the coach takes you out, and you sit there waiting until your next chance. The constant microbiomes are constantly creating new life. The real specialists are inventors, and anabolism is something that we monitor to see where it is going. Interfaces, biotic or abiotic, are deep. We call them also biofilms, if you want, are important. And there you have always then information communication technology. Interfaces often create privileges for their microbiomes. And we can even transplant microbiomes as a whole. The first issues are there. We will have to learn to deal with the services of microbiomes, particularly if we talk about climate change, and I'm saying it will become more important in the future. In terms of manufacturing microbiomes, I see a lot of role for them in protecting us. I'm not against antibiotics and disinfectants, but we overlook this. They are very important in the interfaces between higher and lower organisms, and we will use gene drive systems to improve that. And they are absolutely of predominant value in making the cyclic economy up and providing local sustainability. And finally, and last, the manufacturing microbiome, it's all based on trade. And through this trade, we get constantly new microbiomes, new processes and products. And that reminds us of indeed what we live all the time. That is our market economy. So again, and as I said in the beginning, it's all very similar. The market economy we all live in, and the market economy which we call microbiomes, and which the microorganisms live around us. Happy are those who start at this moment in microbial resource management, because it's a whole new world that is opening up. And these are my co-authors. Thank you.
Very much. The lecture is open for questions. I would like to ask one question about microbiomes and their industrial usage. I mean, to keep, let's say, a pure culture in good shape for industrial use, it's very, very difficult sometimes, and you know, things happen, bad things. How do you view the possibility in the future of keeping a microbiome healthy and active for, let's say, productive work? You can use the microphone.
This is a very good question.
I'm glad you said that.
Yeah, thank you. I alluded to it. The point is, they are always at the best. I've never seen a microbiome go without activity, but it can change activity. So that's my problem. It can do what I don't want to do. It evolves in the wrong direction. Instead of producing X, it produces Y, because they have found a way to take more profit from Y. And I'd rather have X. And then I must try to curb it. That's the problem. But in terms of activity, they constantly evolve. It's like us. You set 10 people together and you say, you make a business. Well, pretty soon somebody will make a side business. And yeah, if this is not in your best interest, you have to, to cut him. And that's the problem with microbiomes. So that's why industrially, lots of people frown. There are two problems industrially. One, you cannot patent it. At this moment, a patent has to have every element that is in it known. And you have there always organisms which are not fully known. So the patenting is difficult, although there are now the first attempts to change the patent laws so that microbiomes will be patented, particularly for gastrointestinal improvement. But absolutely sure that they do what you want. No, they are intelligent. They are just like you hire a very clever person in your team, in your multinational, and then, yeah, you have to keep him loyal. And if he is disloyal and goes his own way, that's the fate of human resource management. That's the problem.
We take human microbes as a microbiome factory. Yes. How we can improve its efficiency for cellulose digestion or mixture of bacteria, protozoa, and
I think this is a Nobel Prize winning question. Huh? If I can resolve it, the rumen, there have been so many attempts. The first real attempt that we have to make, and it has been done in Australia, is can we avoid that this 5–10% methane keeps escaping? And I was indicating this. And the answer so far, the Australians were looking at some combination with the rumen cell wall, where they then put in some antibiotic type of production against those methanogens. I don't think you can work negatively in the rumen. In the microbiome, you have always to think positively and say, can I put into the host something which makes use of that methane? And then we are in the right direction. So the indication that I gave was put into the rumen environment, into the mucosa, organisms which do something better than making methane, which the host profits from, and then it will happen. And that's probably the line that is now going. Make butyrate producers in the rumen mucosa. This is one of the lines that are now explored. If it will work, that's another story. But the main point is think positively. You have to handle it, sir. Yeah.
Okay, we'll have quick questions and hopefully
Quick answers.
quick answers. I wasn't going to say.
Thank you very much. I was excited yesterday when you were talking about the microbiome. And I saw you as a microbial ecologist. And today you have gone further to excite me on this aspect of microbiology. I'm an environmentalist. And I come from a background where we have a lot of pollution, crude oil pollution. My first paper is, although not directly on the pollution, crude oil pollution, is on environment. And my second paper touches on what you now, although we had an idea of it, like if I had known of this before now, I would have brought it into my work. Allowing nature to take care of itself and see what the natural environment could do in terms of biodegradation of wastes. My second paper is on compost degradation. And let me go back to crude oil. I'm a consultant, and I also appear in court to defend microbiology. The problem is, yes, microbes can biodegrade, bringing into play your idea of microbiome. But we use the word consortia, working at oil-water interface to cause biodegradation by removing surface tension and all that. Now, the non-microbiologists, like when I'm in court, they argue that how can microorganisms degrade crude oil, that is petroleum hydrocarbon? And we say in literature worldwide that microorganisms in the environment can degrade crude oil given ample time. So I think that is where you are going to. I'm talking about microbiome, okay. A consortium of organisms acting together to bring about a result.
But the first rule is it's all about change of free energy, so the thermodynamics prevail. It's all about business. If there is no business, if there is no money to make, microbes are not there, microbiomes are not there. So you must first define the business case. I'll explain, but we have to discuss this later. But
yes, we have to.
I'll be very brave. If there is no oxygen or no high-value electron acceptor, oil will not be degraded. Otherwise, it wouldn't be there down in the subsoil. The moment you bring in oil and you bring in oxygen or an alternative oxygen, and then you bring in all the wisdom that exists in nature, that means all the different players, the microbiome starts to form and they burn your oil quickly. But you must first create the case. but I'll discuss that later with you.
You know, why was it not applied in the last
Oh, it is applied. It's applied,
oil,
it's applied.
oil spill?
We learn every day to apply it better.
Well, we see.
Question? Okay, can we have a quick question, please? We're going to,
Yeah, it's finished then. Just one.
we're already way over time. If you want to stay till 7, 8 at night, and then have the poster session, keep asking questions, this is fine. But she was first.
Microbiome of the gut.
Yeah.
Is there a relationship with the autism and colon cancer?
At this moment, this is a good question. I will answer. Everything, almost everything now is explained by the gut microbiome. Obesity, autism. Is it? I am not a medical doctor. I read and I observe that a number of cases are clearly established, others are still doubtful. That this is a lot that in our, this few kilograms that we have, that there are so many genomes interacting with us. This is very clear, but I think we should wait more evidence from the medical field. Thank you. Thank you very much.